Hi, I'm Josh Kane, and I'd like to present to you our recently published work, Lineage-Specific Viral Hijacking of Non-Canonical E3 Ubiquitin Ligase Cofactors in the Evolution of VIF Anti-ApoBec3 Activity. Human Immunodeficiency Virus 1, or HIV-1, uses the viral accessory factor VIF to overcome a host block to viral replication, the antiviral ApoBec3, or A3, proteins. In the absence of VIF, A3 proteins from infected cells are packaged into budding HIV-1 virions, and in the next cycle of infection, these packaged A3 proteins antagonize reverse transcription of the viral genome, greatly reducing HIV-1 infectivity. VIF counteracts A3 activity by acting as a substrate adapter between A3 proteins and a host colon-ring ubiquitin ligase complex consisting of Col5, ELO-B, ELO-C, and RBX2. The VIF hijack complex catalyzes the polyubiquitination of host A3 proteins, leading to their degradation via the 26S proteasome. This VIF-mediated depletion of A3 proteins from infected cells rescues HIV-1 infectivity. Our group and others recently discovered that VIF also recruits the transcription cofactor CBF-beta to the hijacked ubiquitin ligase complex. As CBF-beta is not known to associate with these ubiquitin ligase complexes, we refer to it in this role as a non-canonical cofactor. CBF-beta normally forms a heterodimer with RUNX transcription factors, which stabilizes the RUNX proteins and promotes their binding to DNA. Our group at the time of this discovery hypothesized that, and later discovered evidence for, VIF antagonizing RUNX-mediated gene expression by sequestering CBF-beta away from RUNX proteins, a dual hijack by virtue of VIF simultaneously antagonizing both A3 and RUNX proteins using the same hijacked ubiquitin ligase complex. HIV-1 is the most well-known and well-studied member of the lentivirus genus, which contains viruses that infect a wide range of mammalian hosts. We selected VIF proteins from 5 different lentiviral clades: the human-infecting HIV-1, the rhesus macaque-infecting SIV-MAC, the cat-infecting FIV, the cow-infecting BIV, and the sheep-infecting MVV. This allowed us to ask the questions: 1. Do all of these VIF proteins perform a dual hijack with CBF-beta? And 2. If not, what, if any, non-canonical host cofactors do they require? To answer the first question, whether or not the VIF-CBF-beta interaction is conserved, we affinity-tagged VIF proteins, expressed them in cell lines, affinity-purified the tagged VIF proteins, and identified co-purifying host factors using LC-MS-MS mass spectrometry. We discovered that while all 5 VIF proteins we examined interact with ubiquitin ligase factors, we observed CBF-beta interacting only with VIF proteins from primate-infecting lentiviruses, those from HIV-1 and SIV-MAC. We also challenged these VIF proteins to degrade A3 proteins from their natural hosts in the presence and absence of CBF-beta. Consistent with the interaction data, we only observe VIF dependence on CBF-beta from lentiviruses that infect primates. To answer our next question, if the VIF proteins that do not interact with CBF-beta require a different non-canonical cofactor, we attempted to reconstitute the VIF hijacked complexes in vitro. We had shown previously that HIV-1 VIF aggregates when attempting to reconstitute a complex with ELO-B and ELO-C alone. Only when CBF-beta is added does HIV-1 VIF form a stable, non-aggregating complex. To our surprise, the cow-infecting BIV VIFs protein formed a stable complex with ELO-B and ELO-C alone, and the reconstituted complex exhibited ubiquitination activity against purified cow A3 protein. We concluded that the BIV VIF protein does not require a non-canonical cofactor. Curiously, the other 2 non-CBF-beta interacting VIF proteins we tested, from sheep-infecting MVV and cat-infecting FIV, aggregated with ELO-B and ELO-C alone. We suspected that these VIF proteins required a non-canonical cofactor that is not CBF-beta. While we were unable to find one for FIV, examining the interaction data for MVV VIF, we discovered a robust and specific interaction between MVV VIF and the host factor cyclophilin A, or CYP-A. We repeated the MVV VIF reconstitution assay, including CYP-A, and observed stable complex formation and we also observed ubiquitination activity against purified sheep A3 protein. We identified a potential binding site in MVV-VIF involving prolines 21 and 24. When we mutated these residues, we observed a loss of CYP-A binding. We also observed a loss of A3 degradation activity in this mutant. When these mutations were introduced into the virus itself, we observed a loss of MVV infectivity in sheep cells, similar to that of a VIF-deleted virus. We also tested MVV-VIF A3 degradation activity in CYP-A knockout cells. We observed that MVV-VIF can only degrade sheep A3 proteins when we rescue the knockout with exogenous CYP-A. In contrast, we observe HIV-1 VIF mediating the degradation of human A3 proteins in the absence of CYP-A, confirming its specific importance for MVV-VIF function. We now have a more robust map of the functional interactions of VIF across the lentivirus genus. While VIF proteins from primate-infecting lentiviruses HIV-1 and SIV-MAC require the non-canonical cofactor CBF-beta for function, we observed at least one VIF protein tested from cow-infecting BIV requiring no non-canonical cofactor, and that the VIF protein from the sheep-infecting MVV requires a completely different factor, CYP-A, for activity. Future investigations will assess whether the interaction between MVV-VIF and CYP-A evolved to mediate a dual hijack similar to that of HIV-1 VIF and CBF-beta. To learn more about this study, please read our recent publication in Cell Reports.